Today we are going to discuss a topic from the chapter Lipid, that is classification of lipid. To understand this classification of lipid, we need to understand definition of two molecules. First is fatty acid and second is lipid. What is fatty acid? Fatty acid is basically a carbon chain with a acid. The carbon can vary in number, so it is written as RCOOH. It can be 6 carbon, 7 carbon, 10 carbon, 12 carbon. Now coming to lipid. Lipid is basically fatty acid plus alcohol, or I can say in simple word, it is ester of fatty acid. Lipid is a ester of fatty acid. Now we are saying that to make a lipid we require fatty acid plus alcohol. In this chapter lipid, we will use basically two types of alcohol. Either we will use glycerol or we will use sphingosine to make various type of lipid. So these are the two basic terms that you should know, and right now the important one is the definition of lipid. What is lipid is, it is fatty acid plus alcohol, or we can say ester of fatty acid. From this concept, if we derive the definition, that comes out to be lipid is a mixture of heterogeneous groups such as fatty acid and alcohol. Is a mixture of heterogeneous groups which makes a water insoluble molecule. That will be called as lipid. Now if we go into the detailed classification of lipid is mainly divided into three categories depending on their constituents. First is called simple lipid. Second category is complex lipid, and third is derived lipid. The definition of simple lipid will be, there will be fatty acid and alcohol. Fatty acid and alcohol will make a simple lipid. What is complex lipid? Is fatty acid plus alcohol plus there will be one additional group. Anything extra will be there, so I am writing this as any group. This will be called as complex lipid. Derived lipid, it is basically a hydrolysis product of lipid. Means from the first two categories, if we do hydrolysis and whatever water insoluble molecule we will generate, these water insoluble molecules will be called as derived lipid. Now we will go into detail of this first category, that is simple lipid. The simple lipid, the basic constituents, as we know, it will be fatty acid plus alcohol. Depending on this alcohol group, simple lipid can be of two types. First is called as a fat or oil. Second is called wax. This fat and oil, the example of fat and oil is triglyceride. Meaning of triglyceride is there will be 3 fatty acid and the alcohol part will be glycerol. So it is fatty acid plus alcohol. So it is called triglyceride. This triglycerides makes maximum proportion of adipose tissue. What is wax? The constituent of the wax will be fatty acid plus high molecular weight alcohol. High molecular weight alcohol other than glycerol. So if there is any other alcohol there apart from glycerol will be called as wax. The example of wax is beeswax, lanolin, so this was the first category. Now coming to the second category, that is complex lipid. So the constituents are important question. The constituent of the complex lipid will be fatty acid plus alcohol plus any extra group will be there. Now these any extra group can be different ones. For example, any extra group can be a phosphate group, it can be a carbohydrate, it can be a protein. So we can add any other extra group. Now depending on the extra group, the complex lipid can be of different types. If we are using fatty acid plus alcohol plus phosphate as extra group, the complex lipid that will generate is called as phospholipid. If we add carbohydrate as an extra group, it will be called as glycolipid. If we add protein as an extra group, it will be called as lipoprotein. So now we will discuss these three molecules in detail and their examples. First we will start with phospholipid. The first thing that we should be able to answer till this point is what are the constituents of phospholipid will be. The three constituents that will be there is fatty acid plus alcohol plus the third additional group that will be phosphate. We know that we have different type of alcohols, and the two main alcohol that we can use in this chapter, either we can use glycerol as our alcohol or we can use sphingosine. If I use glycerol as my alcohol, means fatty acid plus glycerol plus phosphate, it will be called as glycerophospholipid. If I use sphingosine as my alcohol, so it will be fatty acid plus sphingosine plus phosphate, it will be called as sphingophospholipid. This glycerophospholipid example, the first example glycerophospholipid is lecithin. It is also called as phosphatidylcholine. The points that you should know from this lecithin, it makes maximum part of cell membrane, or we can say it is the most common lipid that is found in the cell membrane. There is a derivative of this phosphatidylcholine that is called as dipalmitoyl lecithin. It is a constituent of surfactant found in lung, secreted by type 2 pneumocytes. So this is the first example of glycerophospholipid. The second example is cardiolipin. Coming to sphingophospholipid. The sphingophospholipid The basic constituents are fatty acid, sphingosine and phosphate. Example of sphingophospholipid is sphingomyelin. Sphingomyelin is a molecule which makes the coverings of the neurons. The enzyme that is required to digest sphingomyelin is sphingomyelinase. If there is deficiency of this enzyme, it leads to a disorder that is called as Niemann-Pick disease. So this was the first category, that was phospholipid. Now we'll discuss glycolipid, the second example of complex lipid. So the basic constituent of the glycolipid molecule will be fatty acid plus alcohol plus carbohydrate. The alcohol that we are going to use in this glycolipid is sphingosine. So first of all, fatty acid will unite with this sphingosine, and when these two molecules will unite, it will make a molecule called ceramide. On this ceramide, we can add the third molecule, that is carbohydrate. Now we have different categories of carbohydrate. I can add monosaccharide as my carbohydrate. I can add oligosaccharide as our carbohydrate, or we can add oligosaccharide as our carbohydrate with an additional molecule called as NANA. NANA is N-acetyl neuramininic acid, also called as monosialic acid. So if I add monosaccharide on ceramide, it will be called as cerebroside. If you add oligosaccharide on ceramide as your carbohydrate, it will be called as globoside. And if we add oligosaccharide, NANA and ceramide, it will be called as ganglioside. So these are the three main categories of glycolipid. Now, this monosaccharide, we can use a different type of monosaccharide. Either we can use glucose as our monosaccharide, or we can use galactose. If I add glucose as my monosaccharide, it will be called as glucose cerebroside. If we add galactose, it is called as galactocerebroside. To digest glucocerebroside, we require enzyme called glucosidase or beta glucosidase. If there is deficiency of this enzyme, this molecule will accumulate in the cell and leads to a disorder called Gaucher disease. To digest galactocerebroside, we require enzyme called galactocerebrosidase, alpha and beta. If there is deficiency of alpha galactocerebrosidase, it leads to Fabry disease. And if there is deficiency of beta galactocerebrosidase, it leads to Krabbe's disease. There is no further disease associated with this oligogloboside, so no further classification. Now coming to this ganglioside can be of three types, written as GM1, GM2 and GM3. What is GM1? This GM1 works as cholera toxin receptor in our body. This GM3, it is the simplest possible ganglioside. This GM2 is digested by an enzyme called hexosaminidase A and B. If there is deficiency of A, it is called Tay-Sachs disease. If there is deficiency of A and B both, it is called as Sandoff disease. Now why this is called as GM? The G stands for ganglioside and M stands for monocyalo. G stands for ganglioside and M stands for monocyalo because the constituents are ganglioside and there is one sialic acid. So this is the second example of complex lipid. The first example of complex lipid was phospholipid. The second example is glycolipid. Now going to the third example, that is lipoprotein. The basic constituent of lipoprotein will be fatty acid plus alcohol plus protein. Example of lipoprotein is HDL, LDL, ADL and other lipoproteins. We will discuss these molecules in detail in the later part of this series. So these are the three main complex lipid that we should know. Last and the third type of lipid is derived lipid. As we are seeing, they are the hydrolysis product of lipid. But once you do the hydrolysis, whatever water insoluble molecule that you will generate will belong to this category. So better to say it is hydrolysis product of lipid which are water insoluble. Example of derived lipid is cholesterol. So this was the complete classification of lipid that you should know, beginning from the basic concept what is fatty acid and lipid and the lipid constituents. Depending on that, there are three categories: simple, complex and derived. The simple lipid, there are only two things: fatty acid plus alcohol, and depending on the alcohol it can be fat or it can be a wax. Complex lipid, there will be one molecule extra will be there, either phosphate or carbohydrate or protein. Depending on that they will give the name. And the further examples of the phospholipid, glycolipid and the lipoprotein and their disease association depending on the enzyme deficiency. And the last category was derived lipid, and the example of derived lipid is cholesterol. Thank you.